Hello, everyone. Thank you for joining today. Welcome to our third Earth2Earth seminar. Please visit our website, earth2earthseminar.com, for more information about the series or to nominate a speaker. Earth2Earth is organised by a governing board with representation from UK universities, Nature Geoscience, and the British Geological Survey. Any UK institution that wishes to have a representative on the board should contact us by email. We'll be holding seminars every 2 weeks at this time and date. They are open to everyone, and this lecture will be recorded and with speaker permission will be available on the YouTube channel with closed captioning. Calendar links and further information are on the Earth2Earth seminar website. During seminars, we ask that you follow our code of conduct, which you can find on the Earth2Earth webpage. And for questions, please use the Q&A channel on the bottom of your Zoom window, or if you are watching via YouTube, the chat function. We should have 10 minutes at the end for questions. So now it is my pleasure to introduce today's speaker. Andrew Davies is an associate professor at the University of Rhode Island. Andrew works on reefs using a mixture of natural history, experimentation, and novel technologies to untangle the ecology of these enigmatic habitats. Andrew received his PhD from Queen's University Belfast, where he spent several years investigating the impact of limpets on seaweed canopies. Andrew has also had several postdoctoral positions at the Scottish Association for Marine Science, and was a senior lecturer at Bangor University, UK, before moving to his current position at the University of Rhode Island. So thank you, Andrew, if you please begin.
So hello, everyone. So thank you very much, Leanne, for that introduction. As Leanne said, I moved from the intertidal during my PhD into the deep ocean when I moved into my first postdoc. So it was a real kind of baptism of fire, moving from the relatively easy, accessible, shallow water ecosystems on the shores of Northern Ireland into the middle of the ocean in terms of the Atlantic, and heading down to depths of several thousand meters. So today I'm going to try and give you a glimpse into that deep sea ecosystem. And I'm focusing largely on the mapping and characterizing of these habitats. And you see that I'm using some terminology, the last great wilderness on Earth, to try and encapsulate some of the difficulties that we have with studying these habitats. And I'm going to justify why I call the deep ocean the last great wilderness on Earth as I go through my presentation. So I'll just expand a little bit on who I am. So I'm an ecologist, and I also work across disciplines into physical oceanography. So by all definitions, I'm a biological oceanographer. I work in a lot of different marine ecosystems and habitats, and I spend most of my time working on engineering species. Engineering species are those that make structures largely in the ocean, and those can change conditions for other species. They can enhance biodiversity, they have considerable contribution to the functioning of an ecosystem or area. I'm a multi-disciplinary scientist. I use computational and technical approaches to solve problems and ask questions in these habitats. And most of my work has been done in an international fashion with colleagues from around the world. So since I was based in the UK for the vast majority of my life, in the last 2 years, I moved across over to America to work at the University of Rhode Island. And I was a spousal hire. My wife was a primary hire, and I moved along with my wife. And since then, we've been rebuilding our research group and getting everything going. So the move was relatively easy for me. So I didn't really have that much of a problem because I was so used to working internationally that it was a real easy move. And I'm very open in terms of using open source data, open access, and etc. So one thing that people sometimes notice is I'm profoundly deaf, which is when my voice may sound a little bit rounded. And I can't hear anything, but through the miracle of medical science, I have what's known as a cochlear implant, which is right now beaming the sound from this webinar directly into my audio nerve. So your voices, when you speak and I hear you, will be coming directly from the computer through Bluetooth into my brain in a rather unique fashion. So I'm technically a cyborg, but anyway, we won't go any further in that. So That does lead me to one thing. I just want to say that if we do have hearing impaired people who want to talk about the problems that they face, I am available to discuss. Just email me, because I'm very open to supporting deaf people who are trying to make careers in academia. So please do contact me if you want advice, or if you know of hearing impaired students or postdocs who may benefit from advice, please do share my contact details. I'm very open to talking about the problems that deaf people face in science. So, the greatest wilderness on Earth. So when I say the greatest wilderness, many of you will think about things like deserts, barren, lifeless ecosystems. Some people may also even consider tropical rainforests to be the greatest wilderness due to the hyperdiversity that we see in these systems. And there's just so much left to discover. Other people will make a claim for the polar regions as being the greatest wilderness on Earth because of the difficulties that human explorers have in accessing these systems. But I will present a different view. I will present the view of the ocean from space and ask the simple question, why do we call this planet Earth when it is quite obviously planet ocean? And that was a quote from Arthur C. Clarke, who is a science fiction visionary who wrote books such as 2001: A Space Odyssey. If you look at a picture like this blue marble image here, it really does strike home at the vast expanse that the ocean really is, and of just how large it is. We can put that into numbers if we start thinking about the sheer surface area that the ocean covers on our planet, 71%. And if we look at that 71% and we break it down into the definition of a deep ocean is defined as anything below 200 meters water depth. 91% of the ocean is actually deep. So it's a huge, huge area. Some of these numbers about the proportion that's been explored, they're somewhat changing and improving every single day. So the baseline was 5% of the deep ocean has been explored, and a minute proportion of that in any great detail. That changed with more recent efforts to improve mapping in this system. We start to see a lot more of the surface area of the ocean floor being mapped. But of that tiny proportion that we have explored, we have uncovered approximately 28 enigmatic habitats, from vents through to cold seeps, through to coral reefs, sponge reefs, amongst others. So it is an incredible area on the planet with just so much that we don't know, based on the pure weight of the fact that we have yet to explore the vast majority of it. Everybody's heard this. We know more about the moon, we know more about space than we do about our oceans. And that number is actually true. If you just look at it at pure mapping, the resolution of mapping that we have of the sea floor, and compare that with what we have of the moon or even Mars, is very, very, very poor. So we have definitely explored some planetary objects in our solar system at a far greater detail than we have the deep ocean, if you just take the pure resolution of mapping at face value. So it really is. It's not a cliche, it's actually true. So I'm just going to take you on a little journey. So we're going to just, the deep ocean, how deep is it, what lives there, and I've put together some cartoony kind of graphics. So we see in the surface waters of the ocean, sunlight rarely travels below 200 meters water depth. Usable photosynthetic light is usually restrained in the top 50 meters or so of the ocean. But that fuels vast ecosystems throughout the entire water column. So we see huge amounts of food being produced by primary productivity in the surface. And that then ends up feeding whole food chains, which yields huge ecosystems like coral reefs, that yields the infaunal systems that we see on the seafloor in sand and mud habitat. So we see some typical depths here as we go down. So the largest cold water coral reef in the world is found between 300 to 500 meters water depth, which is about the equivalent of a typical submarine. We see blue whales diving to 110 meters. As we go down to 1,000 meters of the ocean, we have no usable light from the surface. Bioluminescence becomes a predominant mechanism for predation, communication, defense. Sponges tend to be found in deeper waters. We also see hydrothermal vents at these depths, where we have seeping fluids coming from the seafloor at temperature up to 400 degrees Celsius, which is an absolutely extreme habitat. Animals and the adaptations that they have become even more extreme. So if anybody's ever watched the Blue Planet Deep Sea episode, I would highly recommend watching that. It's absolutely incredible to see the adaptations that people have captured on camera. You know, things from teeth, from huge jaws that can extend through to bioluminescence. So as we get into the abyss, we're reaching depths of 4,000 meters. The vast majority of the deep ocean floor is covered in sand or mud, and it looks relatively featureless. However, recent studies have actually shown that although it looks pretty flat when we look at the mapping data that we do have, when we start to look at this context over larger areas, the deep sea floor is actually covered in huge amounts of hills, knolls, undersea mountains known as seamounts that can rise from the seafloor. So there is a lot of topographical variability in the abyssal zone. It's not completely flat. And that variability can also yield huge amounts of organisms that live inside the sediment. So there were studies done in the 1970s which suggest that the deep ocean sea floor was amongst the most hyperdiverse parts of our planet. Far, far more diverse than tropical rainforests, but they did that in a highly productive zone. The vast majority of the ocean does have a good level of species, but they tend to be clustered around areas where there's strong food supply and favorable conditions. The deepest part of our ocean is known as the Challenger Deep. And this is where only 4 people have actually visited that deep part of our ocean. Recently, we've had Victor Vescovo, who headed down in about the last year and was looking at these habitats. We've had James Cameron, the director, producer of Avatar, and then the first 2 pioneers of Jack Picard and Don Wolfe, who used the bathysphere with the picture that I showed in the bottom right-hand corner of this PowerPoint slide. So it really is undiscovered. There's very few people have actually transited into the deepest parts of our ocean. And it goes down to approximately 12,000 meters deep. Challenger Deep is 11,035 meters. So there's lots of stuff living there. There's lots of things yet to be discovered, and there's relatively few people have actually been. Much of our deep sea floor does, in all honesty, look like this, okay? Which is pretty boring, I must say, okay? If you look at this, you're thinking, what is this guy talking about? Why is he so excited about a deep ocean if most of it looks like this? We see some sand, some gravel, some evidence of hydrodynamics, you know, with ripples being formed in the sand. And we see some traces of animals here, like these sea urchins. If we were to put a core in, take that core and sieve out the sediment and leave only behind the animals, we find a huge amount of infauna, animals that live inside the sediment. And it's very diverse. And these critters do lots of different things. They move productivity, which has landed on the deep sea floor, into the sediment. They rework it. They move oxygen around. They're doing lots of different things. They're very, very interesting, and there's a lot of them. But today I'm going to spend time talking about more enigmatic systems, such as cold-water coral reefs. This here is a picture of Lophelia pertusa, which is known as the white coal. The bright white on the outer side, on the left-hand side of the slide, is the living coal. And you can see the polyps emerging from the skeletal structure. The darker part in the middle is known as dead coal. And this is where the coal polyps are no longer alive. And it's been taken over by Biodoran's hydroids, boron sponges, and other species, all living within the structure. Living coral in itself is not terribly diverse. It's a bad thing for a species that's a reef former to yield to other species. It wants to dominate the system in order to further its growth. But the oftentimes partially decayed reef structure is very, very diverse. And that's what you see in this picture with these 2 extremes of the level of diversity changes quite dramatically from the living coal as we move into the dead coal. So these are reef formers. They build huge structures which can be more than double the size of your house in elevation, and they can extend for many hundreds of meters. 2 years ago, we discovered a new reef off the coast of the southeast US that extended for many hundreds of kilometers. And they're ginormous structures that contribute so much to the functioning of the ocean. Yet we've not yet been able to capture that and quantify it. There are other types of corals, such as solitary corals. These are ones that grow largely on their own and are in a reef-like structure, but they may be in an aggregation. So they will find them oftentimes growing alongside other individuals of solitary corals. This is known as the Bubblegum Coal, and that's pretty self-explanatory as to why. You can see the bubblegum shape and the color, the pinks. They're also white, orange as well. And this is very common in the North Atlantic. We find it in the northern latitudes. That's solitary coal. We also have sponges as well. Sponge grounds where we have large abundances of sponges, whether they're glass sponges or demo sponges. They grow on seamounts and banks and so on. And we see these large aggregations. Again, hyperdiverse system. So I'm just going to play you a video from one of our recent projects, which is known as sponges. It may not play too well, but hopefully you'll get some footage out of it. I'll narrate it. This is from the GeoSTARS. This is 250 meters, 300 meters, part of the Sponges program, which was the Horizon 2020 project that is due to finish in December. And these are a mixture of corals and sponges. You can see the bubblegum solitary corals emerging from smaller reef structures of Raphelia, and you can also see some sponges interspersed, both glass sponges and demo sponges. And you can see the passage of the fish just swimming around, plus also the particulates in the water column, showing just how productive these habitats are. It's actually receiving a huge amount of food from the surface. That's what's fueling these systems. And you can just see the amount of fish and other organisms swimming around. It's a very, very diverse, very productive system. So lots of life and lots of things happening. So one of the big challenges that we have in the vast expanse that the ocean is, and the deep ocean in particular, is that we do not really know where things are. And that's what I work on and what I focus on in my research group. So we can look at vast databases like Orbis, which has well over 50 million observations of species distributions from the ocean. And we can look at, I've got two different groups of organisms here. I have the cnidarians, which include corals, and then I have peripera, which are sponges. I've subset the entire Orbis data set down to any observations of that particular group of organisms to below 200 meters water depth. You can see that for cnidaria, we're missing huge amounts of the Indian and Pacific Oceans. We don't know much about what's there. There's been relatively little discovery, relatively little sampling effort. And you see that a lot of the kind of brighter colors, like the aqua and the greens and yellows, are clustered around wealthy, well-developed countries. So our sampling effort is very much biased towards countries with huge amount of resources, whether it's access to oceanographic vessels or whether it's financial. They have the ability to get out there and sample a lot. In every other part of the world, it's quite depauperate in terms of effort. And it's particularly pronounced for peripheral. So we haven't had that much in terms of sampling effort and survey around the world. When we start looking at pure mapping, just pure multi-beam mapping, where we're using research vessels or other ships to collect kind of sound images of the sea floor to build an understanding of the structure of the sea floor. And you can see I've taken all of the data that I could get my hands on from freely available data sets, and I've focused it on the Pacific Ocean here to show you just how little broad scale mapping we actually have. If you compare this to the moon, we have 100% coverage of the moon in terms of mapping. When we look at the Pacific Ocean, we have very little. And it's clustered again around well-developed countries like the US. You can see the Hawaiian Islands as a major transit point. Research vessels constantly mapping on their way to Hawaii and back again, and so on. So we do have this issue with lack of comprehensive data for much of the deep ocean. So how can we actually do anything in a system, and what tools do we have? The tools I specialize in is the use of species distribution models, which all of these words that you see in this slide basically describe the same thing. Okay, so if you ever come across a paper talking about habitat suitability models, these are all the same thing. They're a suite of models that allow us to take in multiple pieces of data, we put them into a mathematical framework, and we can try to predict where we can find organisms in systems where we may not have the best data. So I focus on this group of models, the correlative models. We take in known species observations, whether it's presences, absences. We also take in abundances and biomass data, which are far, far rarer than just pure presence. We have huge amounts of presence data, but not much in terms of quantification data for the abundance and biomass of deep ocean species. We then couple that with environmental or geophysical data. So that may be deep sea mapping, it may be extrapolated remote sensing data that covers most of the planet, bathymetric data products such as the GEBCO dataset or the SRTM dataset, which are broad scale, global scale representations of the deep ocean floor in terms of the map. And we can then put that into the mathematical framework of all of these algorithms and different types of models. So MaxScent is a very common model which was popularized in 2006 and is probably the most used species distribution model. Really because there's a very nice user interface for it. So people are using this quite a lot. It's a very low barrier to entry. When you start going into the machine learning algorithms like boosted regression trees, random forest, the barrier to entry starts to increase. So we're using languages like R, Python, in order to build computer programs that run this kind of model. And of course, we have the older statistical models, such as generalized additive models and generalized linear models, which are still utilized today in developing correlative species distribution models. Now we make the model, then we have a whole load of different steps that we need to do in order to understand the models that we produce. We have to do basic diagnostics, performance testing, we need to look at the contributions of variables, and we also need to focus on producing estimates of uncertainty, which is one thing that until recently was missing from a lot of species distribution model projects, in that we could not really say how uncertain our predictions were. So if you're a manager and you're using these predictions to help inform, let's say, fisheries policy or the designation of marine protected areas, you need to have an understanding of the uncertainty that's inherent in the whole of the model, that's communicated in a way that makes sense for them. And that has been lacking until recently. And we've been working on trying to improve that in our more recent outputs. We can produce it, put it into a package that can go online, it can be communicated in reports or papers. So species distribution models, in the time that I've been working on them, has practically exploded in the amount of use, particularly in marine science. So they've really found a tremendous foothold, this whole group of tools. So I did a Web of Science Scopus assessment where I just looked for some keywords of ocean and species distribution model or habitat suitability model, two main terms. And then this is what I saw. So we can see huge amounts of studies using these tools now, almost exponential relationship. And they're being used in a variety of different things. You know, trying to understand species distribution, the management, conservation efforts, but also better understanding their niche. And that's one real big knowledge gap that we have for many oceanic species, particularly in deep ocean, is that we don't really know what the conditions are where they live. We can guess it for sure. And also we're seeing increased uptake in using species distribution models to try to predict responses to change, climate change, for example, or fishing efforts, trawling, and so on. So here are some examples from different models that I've pulled from the literature and how people have been using them, not just in the deep ocean, but also in shallower waters as well. So we have spatial planning, we have the designation of essential habitats, so trying to identify where essential habitats are for commercially important species. We're also looking for risks in diseases, which is an emerging area of research, particularly over the last decade or so, and trying to understand how disease patterns are occurring in the ocean. And the people have been using them to try and better understand the impacts, for example, of fishing activity on species. What I tend to work on is in the top right, which is the distribution of species, trying to produce maps that help us better understand the biogeographic distribution of different species. And then this climate change example here is looking at projecting changes into the future to see how that may influence species distribution. So it's a huge area of research and lots of different players all developing different aspects to this one, basically developing from this one tool, which has been popularized over the last couple of decades. So here's an example of one of my older models, which is Northeast Atlantic, for the white coral that we saw in the picture earlier on, which is Lophelia pertusa, and trying to predict where these species are found. And if you imagine that we do not really have 100% coverage, we do not know that much about where the species is found in many parts of this map. So we really want to use these tools to help inform us. These are great tools because they show the red here, shows where the habitat is potentially highly suitable. It does not mean that we will find that species there. It means based on our understanding of the niche of the species and the environmental data that we have for each of the grid cells in the model, that the conditions are likely for that species. Does not mean that we will find them there, because when we go there, it could be that our data is not terribly good for that particular location. So we may have mischaracterized it in terms of temperature, topography, sediment type, or so on. It may also be that there are biogeographic barriers that are stopping the species from colonizing that region, such as larval dispersal. It may be that we do not have a critical missing data set. And one of the biggest critical missing data sets for the deep ocean is sediment and rock distribution. We just do not have very good data that extends across the whole of the ocean. It's not continuous, it's collected in very few areas. So oftentimes we see maps like these with a huge amount of red, which is definitely not the true distribution of the species. So some of the avenues that we've explored to try to improve these kind of models is to use better data. So on the right-hand side, you see some examples of different models that I've generated for a canyon. So the previous one, this one here, is this one in the bottom right here. Okay. So you can see that this canyon with the contours that I've outlined is predicted as being moderately suitable habitat for hard corals. It's being predicted as being very suitable for soft corals, solitary corals. But you can see that it predicts the entire canyon as being suitable, which is not great if you're a manager, because we are really providing the view that we need to, if we want to say, let's protect this canyon, let's protect the corals in the canyon, we have to shut down the entire canyon to fishing, which can be a problem because people do rely upon fishing for their livelihood. So we are dramatically over-predicting the potential habitat for this species. As we start to kind of improve the model by using higher resolution environmental data, we get into 370-meter grid cell size. So each one of these grid cell size. This is a kilometer, 1,000 meters per grid cell size. This one here is 370, and just make out the size of the grid cells from the differences in colors. You can see that our prediction becomes more constrained. So we can highlight the two walls of the canyon as being potentially suitable for corals. Hard corals, we have this kind of weirdly prediction down the center axis of the canyon, which is primarily soft sediment. So you're not going to find that many hard corals and soft sediment. So that's an example of missing data. So temperature, salinity, it's all good. But missing data is that we don't have any sediment data in here. So we predicted this central axis. If we go up to even higher resolution, we went out and we did a specific survey of the canyon using high-resolution multi-beam, achieving resolutions of 10 meters. You can see that we start to constrain our prediction even further. We start to predict certain axes of the walls of the canyon as being suitable. So it becomes more useful. And people over the years have been really focusing on trying to develop strong local scale models that are really of tremendous value to conservation agencies and managers by constantly iterating and improving their data. Now, that's not to say that the early models that we did are useless. It's to say that started the basis for many of these studies. So my models earlier on said we need to go and look at these areas, at these canyons. So we go and look. We find that they don't support life in what I'm looking for, like corals or so on. That's a good thing. We've actually learned something and we can then iterate it again. So, as I've mentioned, the species distribution models actually have many impacts on, actually have many problems and challenges. So you see that I mentioned about sampling bias towards known areas, wealthy countries, and so on. And it is a major challenge for us when we're working in deep ocean because the expense and cost of going out and working from research vessels can be anything from 25,000 US to 50,000 US a day. That expense is so high that the funding climate doesn't really support true exploration. There's some agencies now which are actually trying to support true exploration of let's go and have a look and see what's there, as opposed to having a directed research question which is built upon previous studies, which is the traditional funding mechanism. So there's definitely some movement toward trying to better understand the distribution of species purely from an observation or mapping process. One thing that species distribution models really like is having some degree of systematic survey. Systematic surveys are extremely rare in the deep ocean because they're very difficult to collect and they require huge amounts of ship time. So it's very difficult to justify. So a lot of the models that we work on and try and characterize where species are rely upon ad hoc, opportunistic data, museum collections, vast databases, and largely they become all about observation data in terms of is it present. Sometimes they never really report absence, and that in fact is very difficult for us to do in species distribution models at the scale in which we work in the ocean, because we haven't truly explored the full area where we have environmental data from. So we can't definitively say that species are absent. And certainly, abundance of biomass data is very rare, but it's improving in quality and it's improving in abundance. So what I'm going to talk a little bit more now is about environmental data. And this is what I've been working on for a very long time, to try to improve the environmental data provision that we have in the ocean. So these are two of the main components where we've seen dramatic improvements so far. The models, the approaches, the access to statistics, technology, the development of R, and the barrier for entry being reduced in R. Tremendous progress has been made in the actual statistical modeling part. The development of machine learning approaches and STEM rules, better computer power, tremendous improvements. Species data, broad-scale data sets like OBIS, GBIF have led to tremendous acquisition potential. So we can get a huge amount of data. But the biggest limit really is in environmental data. And that's where people working in earth science can really help improve, because a lot of people have observation data from sediment substrates and so on, or from bathymetric data in terms of multi-beam mapping, that's not yet openly available. So I can't acquire it to build it in my models. So environmental data is absolutely pivotal. It is the foundation from which all species distribution models are made. So if you have shoddy foundation, as typified by this, you're going to have a problem later on in the line. So environmental data are absolutely important. We build all of our models from it. We want to get the best possible environmental data over the best potential spatial coverage we can. So there are lots of errors and lots of uncertainty that we need to work on. So I'm just going to give you a little bit of an overview of how we've improved recently in terms of acquisition of new environmental data. So of course, multi-beam echo sounders are common now on many research ships, and we're collecting this kind of data at a huge rate. The Ocean X Prize was recently awarded to a company using autonomous vessels to improve mapping. We see a lot more use of ROV technology and also AUV, autonomous underwater vehicles that are able to operate without human piloting. So if we look at marine data, we see it's definitely becoming more available, but there's still more we can do in order to better improve that. So here I've just got two graphs again from searches that I've done online to try and look at how much data is available out there. And you can see Pangea is the database where we can actually download data. It's a bit like the BODC, British Oceanographic Data Center. So Pangea is the German equivalent used by a lot of European projects. And then multi-beam data from NOAA, which is the National Geophysical Data Center. So you can see huge amounts of data is now becoming available relative to earlier on. So we're definitely making great progress here. Here's just a few screenshots of various different resources that are out there if you're interested in actually acquiring this kind of data. GEBCO is the global grid, as well as the SRTM dataset, global bathymetric models, and they're relatively easy to work with, if a little bit large. So, Andy, everything sounds great. So what is the problem? The problem really comes down to the fact that a lot of our development of species distribution models relies upon environmental data that's a little bit, shall we say, not brilliant. And it needs a lot more characterization and a lot more work to better understand the limitations of your data before you go down the pipeline and come up with a prediction for a species. So a lot of studies that I went out, I did a recent survey, I went through different papers to try and look and see, did anybody ask any questions of their environmental data prior to putting it into a whole modeling workflow? And 80% did not. And then I could not replicate every single paper. I could not replicate any of their creation of environmental data. And I could not download that data to test independently. So there's a whole transparency issue at this fundamental level. When we're trying to characterize these habitats, we're producing maps and models. The basis is the environmental data is not terribly transparent. So we're trying to work on trying to improve this down the line. So we have come up with 3 steps towards the gold standard for environmental data. And I presented at multiple conferences and tried to get people thinking about this, stimulate a conversation in terms of trying to make environmental data a bit more important in the whole modeling process. So I'm trying to press the development of validation metrics. I want to improve transparency in methods. And I also want to try to increase the sharing and open access to environmental data, as well as other components of models. So I started this project called GlobEms, which we are trying to develop better environmental data in the world. So if you're interested in this, please do feel free to touch base, and I can talk a little bit more. So SDMs, in some cases, only provide limited insight into our understanding of the environment. So we use these kind of technologies that you see in this slide here to try to collect better data on the niche of the species. I'm going to go through these in a minute, but you can use models in some shape or form to help us understand the context in terms of the environment where the species live. So what temperatures, what current conditions, what oxygen levels do they rely on? We can use models to help inform that. But over in recent years, the last 10 years or so, we've been working quite hard and trying to get better observations of time series. So this here is a Benthic Lander. We drop these over the side of the ship, they sink down to the seafloor, They sit on the sea floor and can monitor conditions depending on your sensor payload. They can also do some kind of experiments if we equip it so. And they take time series measurements of the environment. So we have the frame, which is a large 2-meter high frame. We have these spheres here, which contain vacuum spheres, which allow us to return the Benthic Lander back to the surface. Here we have a weight, which is perfectly synchronized with the lift that we get from the buoyancy spheres. So it sits on the sea floor. We have these 2 releases, which you can just about see the shiny silver, that will release the weight, and then it can return back to the surface when we're ready. We have current meters, we have experiments, and we have other sensors to try and better understand it. So here is some examples of this kind of data. So we have used Benthic Landers to try to capture the environmental conditions for different parts of the world in terms of the reef structures that live there. So here we have an example of different types of data. We have temperature, we have upwards and downwards water movement, and then we have the north-south velocity of the flow in this system here, which is in between the island of Mingulay in the Outer Hebrides and the Scottish mainland. And we've done a lot of studies in this region. We wanted to try to see what was happening in these reef areas, so we put landers down. And we came up with some very interesting mechanisms that were actually occurring. So what happens is, as the tide starts to change, we see a hydraulic jump form, which pushes warmer water and also food supply to the reef. These mechanisms would never ever be picked up by a species distribution model because the resolution of data we have is just not good enough. We do not have hourly minute level data for broad scale areas of the ocean. So we have to resort to different technologies to give us that insight. So here we have an example of food supply mechanisms. In the Gulf of Mexico, we have a different mechanism, which is typified by this DVM, geovertical migration, and the resuspension events near the reef structure itself. So we can see that there's lots of different things happening in reef structures around the world. My final example of using high resolution data is to look at what's happening in canyons. This canyon here is adjacent to the one that I showed the SDM for in the earlier slides. We put several landers down the canyon axis. We wanted to see what was happening in terms of the physical conditions. So the landers are able to give us huge amounts of data. So we have, for example, a backscatter data set from 2 different areas. We have a fluorometer, which is measuring chlorophyll or fresh organic material, which is being delivered from the surface, and turbidity, which is sediment in the water column. So we can see lots of cyclic patterns that are happening, some random, some with incredible severity. There's lots of stuff happening. And again, the point I'm trying to make is that we can have the best environmental data in the world ever, but it's probably not going to cover the time periods that we need in order to really, truly characterize the ecosystems and understand their niche at a level that really gives us the best possible models. So you have to not just use one approach, you have to use all of the tools that you have available in order to build a holistic view of the environment and ecosystem. So in summary, I would say that we are definitely in a golden age of discovery for deep sea ecosystems. There's no doubt. The access to technology that we have is really revolutionizing the way that we approach studying these systems. You just have to look at the availability of autonomous underwater vehicles, recent developments in autonomous ships, autonomous mapping capabilities. Who knows, within 10 years' time, we could have whole fleets of systems out there which are just constantly mapping the seafloor and sending back high resolution, almost real-time data. The big problem is that we do need strong collaboration to work with these systems because they are expensive, technologically complex. So we need ecologists, oceanographers, technologists, all to work together to come up with these kinds of projects that can address broad-scale challenges with trying to understand where things are in the ocean. And the final point really is that the vast majority of our ocean still remains unexplored. And who knows what's out there? Could be anything, you know. Could even find Atlantis. I don't know. There's probably something out there that we've not yet discovered. So thanks very much for listening to me for the last 45 minutes or so. And I'll leave you with this slide from one of the models so you can see the vast expanse of the ocean. And I'll just reassert my offer for anybody who has hearing impairment who would like to talk or share their experiences with me. Please do drop me an email. My email address is on this slide here. Thank you very much, everyone.
Thank you so much, Andy. That was really good, really interesting talk. So we have time for questions. Don't forget to put your questions in the Q&A, or if you're on watching YouTube, on the chat. So the first question we have is, can you use autonomous surface vehicles to aid your studies, from a non-coral researcher?
Well, I mean, it depends what that autonomous surface vehicle is actually going to do. So if that surface vehicle is going to use multi-beam mapping, then definitely we can use that, because we do need better data to describe the topographical variability of the seafloor. So yes, autonomous surface vehicles, yes. If those autonomous surface vehicles could launch a submersible of some description, yes, for sure. We can get pictures, video, and so on. Absolutely. We can also benefit from autonomous surface vehicles in terms of collecting information on surface productivity, which we can then feed into models to estimate carbon export to the seafloor. So there's multiple links, and the answer in the most simple is absolutely yes, 100%.
Cool, thanks. So what environmental sensitivities go into the species models? Are anthropogenic effects like pollution considered?
So no, not really right now. There's been several different types of studies that have used estimates of fishing activity, which is the predominant effect in certain deep sea areas like coral reefs and so on. Bottom trawling is a major threat. That has been used in some models to try to estimate the effect of trawling on biomass, particularly in sponge grounds. So if you look at some research done by the Department of Fisheries and Oceans in Canada, they've produced these kind of models that have used fishing effort. Also, some researchers in Australia have as well. So yes, on that aspect. Pollution, no. Our understanding of how pollution affects deep sea corals is still emerging. There's been, I would say, 6 or 7 studies done on the effects of oil and gas exploration in terms of drill cuttings and contamination. But there's not been that much more done on anthropogenic effects. So we don't have a lot of data.
Okay, cool. Is there the potential for invasive species in the oceans like there is on land? Are there places in the ocean that would be considered overtaken, colonized by different organisms if they were transported there?
So the harsh reality is that there's probably more invasive species in the oceans than around land. So invasive species are a major problem. And I did a study on the Marine Aliens Program, that was 2005. We published a paper in Proceedings in 2010 on the dispersal and spread of marine invasive species, mostly algae, from the points where we first saw them. And you can see them spread throughout the ocean and throughout the coastline. And there are a lot of them, and the potential is huge. Biogeographic barriers, do exist in the ocean. Invasive species are a major problem. And we see invasive species moving about in ballast waters from ships, for example. Fish farming and oyster industries were also a major vector for the movement of invasive species. So yes, there are lots of invasive species.
Just to follow up on that one, somebody's asked, how is an invasive species defined in the marine context?
Good question. So although my presentation wasn't truly on invasive species, I can answer these questions a little bit. So in a marine species, an invasive species is anything that is non-native to that region and has moved in, usually through an anthropogenic activity, or its dispersal has brought it in from an area where it actually exists through time. So let's say examples. One of the biggest invasive species that we have in the UK is Didinum vexillum, which is a tunicate. We also have an invasive algae called Sargassum muticum, which came from Japan. And that was brought over, I think, through the oyster industry. And then it spread from single point locations on the south coast of England, spread throughout some areas on the Atlantic coastline as well. And it's moved up the coast. So essentially, it's anything that's moved in that would not be there if it wasn't for our activity. Natural dispersal in many cases does not mean that a species is invasive. It's if we have brought it there through our activities or via a mechanism by which it would never have ended up there. Case in point is oyster industry, bringing spat from different parts of the world, cultivating that in your waters. You can bring with that invasive species, that you go into a port in some area of the ocean, you fill up your ballast tanks, you go to a different area, you release your ballast tanks, you can spread invasive species.
Thank you. Are there enough data to resolve systematic shifts in species distribution patterns over the last several decades in response to climate change?
Yes, so there's been multiple studies done on this kind of shift in species distribution. We had one paper published this year in Global Change Biology where we've been trying to look at the shift in distribution of corals, fish, and so on, deep sea fish, and what's going to happen. So yes, there is enough data. We are largely relying upon models like data from CMIP 5, CMIP 6. So we are using large-scale global climate models or downscaled regional climate models to achieve that. To look at historic hindcast shift, then yes, that's the potential. People have attempted to look at how deep sea species have moved in the past. But if you look at the amount of data going back, it was really about the 1950s that we see the huge explosion in ECHO. And then recently, I would say in the last 20 years or so, we got that. The missing link is temporal data. So, we don't have a lot of it in terms of observations of species and so on at a temporal resolution that we really need it at. Global satellite monitoring like CRIFT and MODIS been great for us because we have continuous data sets of surface productivity for 20 years or more, which has allowed us to really look at high resolution changes. So I would say 50-50. Yes, there is enough data. Can we do it well? Probably not as well as we want to be able to. So I think there's definitely scope for improvement.
To follow on from that climate change question, is there a chance that environmental data gathered now will have a very short shelf life due to climate change? How long will these data remain useful at the current rate of climate change?
So although conditions may be changing, any data that we have now is of incredible value. So it doesn't matter if the conditions are changing. What's important is that variation. We need to understand variation and change. So I don't think data has a short shelf life. Any data we collect is of value. Whether or not we have the time as a scientist to be able to work with that data is a question. But yeah, it's not having a short shelf life. So if I interpret your question slightly differently, so let's say I take a 20-year annual average value, end up with a single value of temperature for a grid cell in my model, then that average value is very, very coarse. I'm not looking at variation around that mean. I'm just looking at that average value from that 20-year data set that I have. Then yes, so it's going to have a little bit of a short shelf life if that average value is changing. So the point you make is very valuable because we do need to take into account time. There's no doubt about it. And that's one thing that many large-scale species distribution models ignore is time and variation in conditions through time. And that's where the observatories and the landers are a real important component of that whole thing, is to try and give us a bit more of an insight into the variation of conditions through time.
Thank you, Andy. And this one's not specifically on your research, but as a deaf person, are there barriers, challenges to full participation in the scientific community? Have these been a major problem for you or a minor one in achieving your success?
Yeah, so I lost all my hearing. I had 25% hearing since birth, and then I lost all of my hearing when I was about 30 years old. I woke up one day, I came downstairs, I was living with a fellow academic. He started talking to me, and there was something seriously wrong, and I lost all of my hearing overnight. What happened was my tinnitus level, which had always been really, really super bad, I mean, crazy bad, actually was so loud, it overwrote the remaining hearing that I had. So I had two choices. Number one was I went into deaf culture, or I had a cochlear implant. And I would say the pivotal time in my life was having the cochlear implant. Now, I'm a fairly unique deaf person. I was born with very severe hearing loss to a hearing family. And everything that I've ever done has been oral. My family have been oral. I've never really been part of the deaf community. I've always kind of been in the middle gray area. So having a cochlear implant for me was a very easy decision. For people who have profound hearing loss, who have the interaction with the deaf community, having a cochlear implant is a different decision. And it's a different one. Very much so. I don't want to go into that, but it's a different decision if you're in deaf culture. Cochlear implants are frowned upon. They don't like them because it's taking people away from the deaf community and making them artificially hearing. So there's pros and cons either way. So it was an easy decision for me to have the implant. The implant for me it's changed my entire life. So prior to having the cochlear implant, if COVID had hit before I had the implant, I would not have been able to use Zoom. I would not be able to understand Leanne relaying the question to me. I would not be able to teach online. And I would still be doing what I normally do, which is trying to type emails and to collaborate in that fashion, which everybody knows is really slow and not very natural. So collaborating for me has been the biggest barrier and challenges I've had is communication. And, I don't have a phone in my office. I don't use the telephone. I can use Zoom. If I can hear the person, I can get a little bit of a feel for what they're saying. And I can also lip read them. And context is really important. So lip reading, being able to see people, the use of Zoom, the use of Skype has been great. FaceTime, fantastic. But, that was a massive barrier until, just under 10 years ago. And that changed my life. There's a lot of barriers for deaf people in science. And science is oral. Everything you do, lectures are oral. Presentations and conferences are oral. Conference collaboration for me is a nightmare because it's so loud. When you break out, you go from the conference room, you go out, you're having a cup of coffee, people are trying to talk to me, no chance. I'm like, okay, we've got to go out if you want to talk to me. So I've got to create situations that work for me, which may not be what other people want. So if you imagine that you're talking to some stranger and it's like, oh, follow me, come away from the crowd. Okay, it's a little bit weird. And people are not terribly open to that. Plus, I have a tendency to sometimes shout and, I don't know I'm doing it. It just happens. And again, that's weird. So people think it's weird, but it just happens. So there are barriers. So I think it starts at school. It goes through all of that until you're at university. You still have barriers and problems. And it's pervasive throughout your entire life. So it's not just in academia, it's also in your life. Whether or not there have been barriers in terms of discrimination I've experienced in the past, and that's happened with access to platforms and research situations that I've been denied access to. But thankfully, through various disability discrimination acts, widening access, more awareness, people have become a lot less prevalent. So I haven't experienced that much in recent times. It's a challenge. There's lots of problems, and achieving success has been a hard slog. Things are slower for me. It's harder for me to naturally collaborate. So it's oftentimes putting together big grants. As a PI especially is really slow and challenging because I'm having to do everything by email or face-to-face visits, where I would, in pre-COVID times, I would actually drive to people's offices and meet them in order to accelerate that process. But with Zoom and COVID, I must say I've been practicing a lot. I think I'm getting better. So I think things are good. A lot better with the cochlear implant.
Thank you, Andy. It's 4 o'clock now, but there is one more question. Are you happy to take that last one?
Yeah, sure.
So the last question is, how good is the data describing the environmental preferences of marine life?
It's as good as it's going to get until we start really going out there and getting better data. It's okay. The problem is not too much the definition of the word good. Okay? It's about actually understanding what that data is showing and what the limitations of that data are. That's the problem. That's the missing link. All data is good. Any data is good. The question really is, how useful is that data? What are the limitations of that data? If we know that, all data is good. The only bad data is data that we don't truly understand. Okay? So that's the difference. That's how I would interpret that question.
Thank you so much, Andy, for the amazing talk and for answering all those questions. Thank you to you all for coming. And just a reminder that our next seminar is in 2 weeks' time. It will be from Professor Zachary Ross from Caltech, who will be discussing how 3D fault architecture controls the dynamism of earthquake storms. So we'll see you in 2 weeks. Thank you.